Hi, my name is John Patterson, and I'm here to talk today about Roger Kornberg, who was an American scientist who is most famously known for his work on transcription. So, a little bit about his background. Roger Kornberg was born in 1947 in St. Louis, Missouri. He's the oldest of three siblings. He has two other brothers, one who is also a biochemistry researcher and who actually helped Roger with some of his work throughout his career. Another who's an architect who actually designs laboratories and helps kind of design biochemistry labs as well. So as a kid, Roger had interests in music and reading. He played instruments and was big into the musical world. Family was also something that was important to him, and most importantly, his family was the one who got him interested in science. His dad actually won a Nobel Prize in 1949 for his research with biochemistry and physiology and how that related to medicine. And then his mom also worked in the biochemistry field. Um, so as you can see, family was important to him and kind of influenced his desire and his interest for science. A quote that I stumbled upon when I was researching, Roger said, "Science was a part of dinner conversation and an activity in the afternoons and on weekends. Both my parents had fine scientific minds and taught by example how to approach questions and problems in a logical, dispassionate way. Scientific reasoning became second nature. Above all, the joy of science became evident to my brothers and me." So, a little bit about his education and scientific career. Roger studied chemistry at Harvard for his undergraduate work. He graduated in 1967, and then he went on to graduate school at Stanford University, where he got his PhD. Here, he kind of looked at the chemical nature of phospholipids and just how their structure and their movement inside the cell membranes. He looked at the different ways they diffuse throughout the membrane and kind of move, and just the nature of phospholipids. So, interestingly enough, some of the techniques he used, and we'll talk about this later, during his graduate work at Stanford University actually helped him in his research for transcription. So he actually did some postdoctoral work as well. In 1972, he was on the Medical Research Council at Cambridge University, where he was a fellow there. And here he looked at X-rays of chromatid and chromatin structure. He worked alongside Aaron Klug during his time at Cambridge University, and in 1974, he and Aaron Klug were looking at histones and then the chromatin structure and how that related back to just overall processes and how those natures. So he was one of the pioneers in this kind of research, and this happened a lot during his postdoctoral work. And then in 1975, he became a professor at Harvard University in the medical school. And in 1978, he actually went back to Stanford as well, where he currently works as a professor in the medical school, researching and teaching about biological structures and just kind of in that department. So, been around education and science basically his whole career. Kornberg's work. So he's, like I said, he's most famously known for his work on transcription, and most notably, obviously, eukaryotic transcription. And just a little refresher: this is when DNA is converted to RNA, and then he kind of focused in on the molecular mechanisms of transcription and just the initiation and the initiation factors. So that was his work, and he actually ended up winning a Nobel Prize for his findings in 2006 for his work on eukaryotic transcription. His other research kind of includes phospholipid structure, like I said, kind of during his graduate school at Stanford, and then chromatin structure and how that kind of relates back to eukaryotic transcription. So he's been looking a lot kind of intracellular and kind of the molecular mechanisms to behind transcription and other processes. So recently, he's actually been working on chromatin and chromosome coiling and how that relates to gene repression and just how coiling relates to the control of transcription, kind of like what we're talking about in lecture these past couple weeks of the tightly coiled portions of DNA. A lot of that work comes from Kornberg as well in his most recent kind of research. So contributions of Kornberg's work, what did his legacy kind of lead the way to? So one, I mean, obviously it helped us understand transcription in eukaryotes and how that process gets initiated and how that process starts. He looked at DNA polymerase II and the role it plays in eukaryotic transcription. And this kind of opened up the door for molecular mechanisms of transcription and the components and initiation factors that are involved in it. This paved the way for new research in the field of the regulation of transcription. So Kornberg's finding kind of opened the door again for just what do these initiation factors mean and how does repression and activation of transcription kind of happen. With Kornberg's work in 2006, this kind of, again, like I said, opened the door for more research to be made and more findings in the future to be made. And a little bit less of his contributions, but still important when he was working on phospholipids, the role it plays in the cell membranes and how the phospholipids move, just understanding that process. And again, the coiling of chromatin and how that relates back to eukaryotic transcription, either repression or activation of transcription. So what does the explanation, the scientific meaning of Kornberg's work, like what did he do and what does it mean? So again, he kind of looked in at transcription and how it worked. Here you can see the initiation of transcription and the kind of overall process. And then obviously here's where the DNA is being converted to the RNA. And like I said, Kornberg's work kind of focused more in on these transcription factors and how that relates to RNA polymerase and the start of transcription. So transcription, Kornberg worked at the RNA polymerase can actually read through the nucleosome. Again, the nucleosome is kind of this overall structure of what is being transcribed and where is it happening in the cell. These nucleosomes can actually help regulate transcription. Kornberg saw different transcription factors act differently, and they combine to RNA polymerase to activate transcription as well. So here is a picture from Kornberg's Nobel Prize lecture of the nucleosome and RNA polymerase starting this transcription of the DNA that's wrapped up. Here you can see the histones and the chromatin. And it's important to note all of his research was done in yeast cells, but he concluded that this is actually virtually identical to mammalian cells. So this was a big breakthrough for eukaryotic transcription. So the initiation factors he found were A, B, C, D, and E. Factor D was also yeast transcription factor 2D. And then transcription initiation factor B is a multi-subunit protein kinase. And actually, he found phosphorylates or activates RNA polymerase II to allow transcription to happen. So he was able to isolate these two factors pretty easily, and he found the role that these two, B and D, played in initiation of transcription. But one problem he ran into was actually isolating the rest of the factors, because it was so diluted down that he was actually losing the important factors, the other ones that he was trying to find, and there was actually a buildup of inhibitors when he was doing this. So he had to find a new technique to actually do this process. At the beginning, like I said, he was kind of struggling, and he got help from one of his fellows working with him, which his name was Michael H. Sayre. So again, factor B is important because it phosphorylates and activates RNA polymerase II, And Kornberg found out that this was an important mechanism and an important step in the initiation of transcription. So this new method they used to actually isolate the rest of the factors was called the fractionation scheme. Sorry, and this was used to isolate initiation factors A, B, and E. And once he was able to actually isolate these three factors, he found out that A, B, and E work with the other one, transcription factor D, to initiate RNA polymerase II. So these work together in a complex, and when they work together, and when he found out that A, B, and E also helped with the activation of RNA polymerase II, this complex is now able to allow transcription to occur. And then he actually found out the structure of a mediator protein, or a multi-protein complex, as you can see right here, is actually helps transfer the signal from these transcription factors. As you can see here, the activator using the mediator protein to transfer the signal over to RNA polymerase II to tell RNA polymerase II to start transcribing the DNA into RNA. So all of this work Kornberg did, he actually found out that that this was an important mechanism, and we didn't know much before this, but he realized that there was this activator complex with the initiation factors, and then he isolated this mediator and found out that its role was to, be kind of a usher over and allow the signal from the activator to go to RNA polymerase II to tell the cell to start transcribing the DNA into RNA. So another important finding of Roger Kornberg's work was the structure of this transcription machinery, as he liked to call it. So he used kind of a crystallization method of the complex. And this was the same technique he used while studying lipids. So he, again, like I said before, he used some of his knowledge that he used earlier in his career to help him with this finding of the structure of this complex, or these complex of the initiation factors. Sorry. So that first started off with a 2D crystal structure, which ultimately was turned into a 3D crystal structure. And this took a longer set of time. So it started off 2D, and then he had to wait a little bit until it turned into 3D. And then he used the X-ray analysis that he also used with chromatin to read these 3D images. He was able to come up with the structures of RNA polymerase II and the other transcription factors. So as you can see here, RNA polymerase II has a 3 angstrom backbone model. It is also a 10 subunit protein. And there's these jaws that he liked to call them, that grip the DNA downstream of the active site. So as you can see from these pictures, again, from his Nobel Prize lecture, these jaws on the RNA polymerase II grip the DNA and allow transcription to occur. And then you can see the different kind of transcription factors here and the complex of the overall initiation mechanism. Again, here is just a different image of that RNA polymerase II. So all of this work was very groundbreaking for the world of science, and specifically on the role of transcription, because it gave us a better view of how transcription was initiated, the different factors and the roles they play, and how transcription actually was regulated and started in eukaryotes. So Roger Kornberg's work was very important and groundbreaking, and again, just won him a Nobel Prize for it in 2006 for this this just important discovery of how transcription initiated and kind of what the structure of these complexes are. So that's it, and thank you very much, and have a great day.